Okay, this time this WebEx meeting will be done as an electronic makeup class for the tissue engineering part 2. Let's start with tissue engineering. This time we will study the characterization and clinical application of scaffolds in terms of tissue engineering. These are the contents. First, we are going to understand importance of porosity in scaffolds. Second, we are going to study how to characterize the porosity. And third, we will study how the scaffolds can be utilized in terms of artificial skin. Let's go with the general features of scaffolds. Properties of scaffolds are influenced by their characteristic. The characteristic properties are porosity, specific surface area, pore connectivity, and etc. And various characterization techniques such as scanning electron microscopy, porosimetry, micro-CT has been suggested to examine the structure of scaffolds. The properties of scaffolds. Molecular transport is related to angiogenesis and diffusion. Angiogenesis is the development of new blood vessels. And molecular transport, molecules such as oxygen metabolize nutrient signals. These molecular transport are required in cell proliferation, and they requires interconnected pore, the pore network. Inability of diffusion resulted in a cell proliferation only at the surface and necrosis in the inside. When the pore is not connected, the cells attached inside the inner core pore cannot get enough nutrient from the biological system, so they will die by necrosis. But at the surface, the cells will grow, but the cells does not make three-dimensional network at the outside and the inside, resulting in the failure in scaffold based regeneration. These are the structural properties to expect diffusion properties. First, porosity is defined as the pore volume over volume of pore plus scaffolds. Surface area is the total surface area considering the volume or mass of a scaffold. Connectivity means a volume of connected pore over the whole pores, connected pores and blocked pores. Pore size is a mean pore diameter. Wall thickness is average thickness of scaffold. Anisotropy is a heterogeneity of scaffold wall. Anisotropy is the different in a dimension in certain direction. For example, certain wall is very large at one direction and thin in the other direction. We call it anisotropic. Cross section is a cross-sectional area at a certain location. Permeability is a degree of liquid flow through the pore of scaffolds. Okay, let's get into the porosity part. Porosity is related to cell adhesion, diffusion of molecules, and mechanical property of the scaffolds. High porosity is advantageous in cell adhesion, cell proliferation, and molecular transport of nutrient and waste. But the scaffold with high porosity resulted in reduction of physical strength. So the highly porosity scaffolds cannot be utilized in bone or blood vessel applications. Connectivity and permeability. Interconnected pore is required in scaffolds for the cell proliferation and development of tissues. Connection among pores facilitates cell adhesion and proliferation, and permeability is affected by connectivity, size, and distribution of pores. Size is also important. If the size of the pore is too small and the channel size between the pores is too small, some kinds of large nutrients such as proteins cannot be transported by that pores. So it is required to obtain as large pore as possible, but too large pore makes reduction in mechanical property of scaffolds. So there should be some balances. An important factors in scaffold design. First, we consider the shape and physical chemical property of the tissue, of the target tissue, which would be regenerated after scaffold is transplanted. And also we determine the preparation method and design the properties when the target tissue is determined. When we design scaffolds, precise geometry and shape is required. In this case, honeycomb type scaffold is utilized. It is easy to control the interconnected porosity, surface area, and cross sections in honeycomb type scaffolds. It's why this scaffold is utilized and the precise geometry and shaped scaffolds. There also exists irregular shaped scaffolds. It is obtained by bubbling method or nanofiber mesh methods. Bubbling method is like that decomposition of porogen or gas formation or particle leaching makes the pores inside of the scaffold. You already know what's gas formation and particle leaching as we learned it from the ceramic part. And the nanofiber mesh methods. Electrospinning is a kind of nanofiber mesh method, and phase separation is another method of nanofiber mesh methods. This is the SEM image of the nanofiber, the scaffolds made by nanofiber mesh methods. You can see many nanofibers and a cell is attached in between the nanofibers. This is the scaffold, and you can see many pores, although the pores are not regular. Okay, let's think about the characterization methods of the porosity of scaffolds. Characterization theoretically, calculation methods such as unit cube analysis or mass analysis is possible. In experimentally, SEM measurement is utilized for the visualization of the pore structure. And also we can apply mercury porosimeter. In this method, mercury intrusion is dependent on the pore size and pore connectivity, giving rise to the calculation of pore size and pore distribution. Okay, let's think about the first method, unit cube analysis. It's best suited in honeycomb scaffolds. Porosity is expressed in this equation, where VF means volume of materials, and the volume of materials can be calculated with this method. And VA is volume of scaffold cube. And here, the volume VA and VF are calculated in this equation, where D means the diameter of the pillar, L is the length of the pillar, W is the width of pillar, H is scaffold height, the macro size scaffold height, and N1 is number of pillars in a layer, N2 is number of layers in a scaffold. Second, mass analysis is possible. Calculation from mass and density of scaffold. Mass, the gram, density, gram per cubic centimeters. Porosity is expressed by this equation, where Vg is the volume of scaffold, Va is volume of scaffold cube. Vg means the volume of pure material itself without pore distribution, pore contribution. Va is the volume of scaffold itself. All calculation from crystallinity and density of polymers can be applied. The porosity can be can be utilized as can be expressed in this equation where dp chi c dc da dapp are expressed in these terminologies. Okay, the experimental methods such as SEM measurement is maybe the best, although we cannot see the everywhere in the scaffolds, but we can visualize the shape of pores. It's a direct measurement of pore size and pillar thickness. Surface analysis by the secondary electron from electron irradiated samples are the principle of SEMs. This is sample. There comes electron beam. The electron beam radiated samples irradiate secondary electron. And then the secondary electron is very sensitive to the surface topology of samples. By this way, the detectors convert the secondary electron signals to the surface topology of sample. So as you can see here, we can calculate, we can measure the size of the pore here. And also we can measure the thickness of the pillars. In this way, we can calculate the pore size, porosity, wall thickness exactly. But with SEM, we cannot observe every part of the scaffold. So this can be a kind of statistical methods from the sampling. And another experimental method, mercury porosimeter, is an average method. We measure the porosity of the whole sample. We utilize the principle of intrusion of mercury, which has very high surface energy. So mercury gets together by themselves. Mercury does not wet something. But if we put high pressure, mercury get into the pore at certain pressure threshold. By measuring the threshold pressure, we can examine or calculate the porosity of the samples. It's the equation dP equals to 4 gamma cosine theta. D is the pore diameter and P is pressure. And theta is contact angle, which is usually 140 degree, because this 100 degree, 40 degree is generally accepted for mercury solid substrate. And gamma is the surface energy of mercury, and it is already set to 489 per centimeter. So when we measure pressure itself, we can measure the pore diameter. First, vacuum facilitate intrusion on mercury into the pore of scaffolds. And volume of incorporated mercury under the maximum pressure is measured. And all the open pores and pore volume is measured in this method. It's the continued properties. Pressure versus accumulated mercury intrusion is plotted as a curve. When the accumulated mercury intrusion is high, it means the porosity is very high. At maximum pressure, reduction in pressure is observed because of the intrusion of mercury, and extrusion coefficient can be calculated. Advantage of this methods. Various parameters such as pore size, torsion, compressibility, accumulated pore volume, permeability, and pore density, and etc. can be measured. But there are some disadvantages. It is not applied for the pore connectivity, and we cannot measure wall thickness, and we cannot measure anisotropy. And also, very high pressure, about 415 megapascal, is required. And sometimes scaffolds made by metal cannot be utilized because metal often mixed amalgam alloy when they encountered mercury. The measurable pore size distribution is between 1.9 nanometer to 400 micrometer. It's a quite wide range of measurable pores. The other method is flow porosimeter. It's a non-destructive measurement and it is well utilized in filter measurement. First, the sample is fully wet with water, and the sample is sealed in a chamber and subjected to gas pass. Gas passes through the fully wet sample. Above a bubble point, the gas pressure larger, exceeding um, capillary union is formed. I'm sorry, this is larger than capillary union is formed at the largest pore. It's simple. When the sample is wet, the pore is blocked by water, and gas comes. When gas pressure is high, at certain gas pressure, the gas can repel the water molecules to fill the pores. It's like that. It's pressure and flow rate. And in dry state, pressure is proportional to the flow rate. When we adjust high pressure, the gas get through the pore, and the pressure and flow rate is proportional. But when the sample is blocked by waters in the wet curve, at a certain threshold point, the the pressure is not enough to push out the water molecules. But at a certain threshold pressure, it can the gas can push out the water molecules. This is dry curve, and this is half dry curve, and this is wet curve. And the interconnect between wet curve and half dry curve means mean flow pore diameter. And the pore size is determined between bubble point here and the point where dry curve and wet curve meets. This is pore size range. The measurable pore is from 13 nanometer to 500 micrometer. Non-destructive micro-CT can be utilized. By micro-CT, we can get the tomography image of the scaffolds. It shows the scaffold's wall and pore at the same time. The resolution is around 50 micrometer. It's an advantageous to measure the fibrous structures. And the qualitative and quantitative analysis is possible on 3D samples. As we know from the CT images for the clinical investigations, we can also get the three-dimensional image inside and outside of the scaffolds by applying micro-CT. And micro-CT is non-destructive method. So after micro-CT measurement, the scaffold can be directly applied to another analysis or to the transplantation. And the principle is quite simple. X-ray beams irradiate in various directions on a sample in various paths, and it makes 2D images. As you can see here, X-ray goes this way, this way, this way, this way, this way, this way, and they can make some focus here and there at every point, and through the focusing, the structure at a certain part, the X-ray attenuation is high. At certain part, X-ray attenuation is small. Can be mapped by the material and the air part. Okay, this is the requirement of the scaffolds. Biodegradability and resorbability is required for scaffolds. It means that it should not induce inflammation and it should be non-toxic. Usually PLA and PGA or their copolymers such as PLGA are widely utilized as scaffolds because those polymers are the polymers of a natural metabolite. I mean the lactic acid and glycolic acids are natural metabolites and they are the monomers of those polymers. And cell-friendly, cell adhesive, and tissue induction properties are required. So sometimes or frequently, amino acids such as RGD sequence is incorporated for cell adhesion. And also the scaffold should have mechanical properties similar to target tissue. So maintenance of scaffold shape is required until the development of new tissue. As you know, scaffolds should be disappeared after the new tissue is developed. Scaffolds are degraded in physiological state, gradually replaced and gradually should be replaced by ECM. So the scaffold location should be replaced by the tissue. The tissue is assembly of ECM and the collection of cells. Okay, then let's think about artificial tissue. Artificial skin. It's an example of scaffold or artificial tissues. Artificial skin in tissue engineering. Artificial skin is utilized to treat burn, bad sore, or skin ulcer. Recently, skin ulcers are also found from the diabetes. And also, it is utilized in cartilage defect and other various skin diseases. There are several types of artificial skins. Generally, two types, wound dressing and artificial skins. The artificial skin has two types, cell-free artificial skin and cultured cell skins. So this is the pyramid of artificial tissues. Here, this is cell-free skin. These are all artificial skins. There are some commercial products like Integra, produced by Integra Life Sciences. And there also exists a cell-containing skin, and the cell is dermis in this case. The dermagraft trademark from the ATS is an example of dermis-containing skin. And also there also exists a small amount, but there exist dermis plus epidermis skin. The Orcell from Ortec International or Apligraft from Novartis are examples of the skin containing dermis and epidermis together. And first, let's think about the wound dressing. Wound dressing is the most simple one. It is used to treat burn or wound. If we have paper cut, we put something made by silicone. It's one type of wound dressing. It protects wound until skin recovery. And also it prevents moisture leak from the body because when there is a cut, that part is opened, so the wet skin, wet tissue is exposed. So moisture can be evaporated through that wound part. But the wound dressing prevent the moisture leak from body. And also It absorbs exudate from the body. The exudate is a kind of waste liquid from the body, and it prevents infection by germs, infection by the exogenous germs. And then let's go to the cultured skin. It is used to treat broad burn or ulcer, somewhat more serious cases than wound dressing. And the ulcer is often caused by diabetes nowadays. Cells collected from patients or other humans are proliferated. They are collected and they are proliferated and attached on a polymer scaffold. The advantage of a cultured skin is large scale is available and less immune response occurs when the patient's cells were utilized directly. And then let's think about artificial skin without cell. It is used in serious burn or skin defect during surgery. And it is made up of upper silicon layer and lower collagen layer. Here there's no cell, but there exists collagen, the natural polymer. The outer part is made by silicon. It induces regeneration of blood vessels and ligation tissues. The role of artificial skin without cells are wound covering. It acts as a channels for human cells, and it assists tissue recovery, and it absorb and degraded in human body. Integra is one of the example. It's a sponge type product made from bovine collagen. And these are a wide variety of artificial skins. It's a wound dressing. Wound dressing is the most widely utilized one. It has the biggest market. And it is divided into without antimicrobial type and with antimicrobial type. Or sometimes there exists wound dressing having some bioactive molecules. Among without microbial type, we can divide them into artificial materials and complex materials. In artificial materials, there exists membrane type, spongy type, gel type, or sometimes non-woven fabric type. In Korean, it's bujikpo. In membrane type, silicone, nylon, PLA, or polyurethanes are utilized. In sponge type, similar. Silicone, polyurethane, and nylons are utilized. All of them are bioinert and biodegradable polymers. In gel type, PHEMA and PGE or pluronic F127 can be utilized. And in the complex materials, there is a two combination of artificial and artificial natural. In artificial silicone membrane and nylon fabric can be utilized, or PHEMA or PU foam can be utilized. The general things we put for the paper cut or some small cuts are all wound dressings. Or sometimes there exists silicone membrane plus collagen type. This is what we have seen in the previous slides. And also some wound dressing could contain antimicrobials and also some bioactive molecules like growth factors. They facilitate the regeneration of tissues. And these are the artificial skins without cells and with cells, with skin. Without cells is divided into two types, allogenic and xenogenic. Allogenic means 동종, from the same species. Xenogenic is from 이종, from other species. So in allogenic skin, human skin without cells is utilized, and xenogenic skin, porcine skin, pig. Porcine skin is utilized. Or sometimes collagen sponge is utilized, and bovine collagen with sponge shape can be the example of this type. In cultured skin, also there exists allogenic and xenogenic. Allogenics are human cells and xenogenics are from others. And they can be divided into cultured epidermis, dermis, cultured skin. That skin here means stratum corneum, in Korean 각질. Dermis is 진피. Epidermis is 표피. These cells are different and according to the location of the skin. The keratinocyte is the outer part, and then epidermis, and inside there exists dermis. So the name and the type of cultured skin is determined by what cells we choose for the skin cell cultures. Okay, these are summary quizzes. Let's think about why pores is important in scaffolds. I will ask you again, but I will give you the answers. Pores are important in cell adhesion and the molecular transport. And the molecules can be nutrients, either nutrients or the waste. And how can we evaluate? It's evaluate porosity of scaffolds. There are lots and lots of methods. Theoretical methods can be utilized to calculate starting from the dimension of pillars and the apparent size. But sometimes SEM measurements can be utilized to visualize, or mercury porosimeter or flow porosimeter can be utilized to measure the pore size. Number 3, what is the role of artificial skin without cells? Artificial skin without cells protect skins from other exogenous attacks, and they help the angiogenesis and skin regeneration. Okay, let's call it a day. Thank you very much and see you next time.